so today Lindy and I are going to show you a demonstration for Lab 14. So my name is Chelsea Maroff. This is Lindy Gates. I'm an instructor here at UNO for those of you who don't have me in class. And Lindy is a graduate teaching assistant and is also teaching one of the labs this semester for Bio 2. So we have 3 CO2 sensors set up on the table before you, and we also have 3 soil samples. So Dr. Robbins will be going through all of this in the lecture PowerPoint and in that lecture video. So I would suggest watching those first. We just wanted to show you a demonstration because this is probably equipment that if you didn't take Bio 1 at UNO, you maybe have never seen this type of equipment before, or maybe you used it one time, but it's just good to have a little refresher on what the CO2 sensors are and what we're actually doing for this lab today. So today we have 3 soil samples. So you probably saw this in your manual already. So we have cool and dry. So that soil has no moisture added to it. We also have cool and wet in the middle here, which we added some water to just before the lab today. And then we have warm and wet. So this is our little ceramic bulb heat lamp, which is just warming that soil above room temperature. And it gets usually a lot of heat in there. So when we go to use a little bit for our experiment, it stays fairly warm while we're actually doing things with the soil. So we're going to take just a small sample from each of these bins and use it to measure basically the change in CO2 in these different experimental treatments of our soil. So, why would CO2 be accumulating after a few minutes when we put soil into a closed bin? So think about that for just a second if you haven't watched the lecture yet. And there's a reason that we don't just call this dirt, right? Because this is soil that has living things in it, and it has microorganisms, and those microorganisms do exactly the same thing that you and I do. They consume food of some sort in the soil, and then they're going to produce CO2, whereas, plants that you're used to seeing, like trees, are going to use CO2 and produce oxygen. So it's kind of weird to think about that there are these microscopic organisms that are using the same metabolic processes that we are, and they're putting off CO2. And so when we keep them in these enclosed chambers, we're able to measure that change in CO2 to see how much microorganism activity there is in our soil. So we're using our treatments temperature and moisture, and we're using them together on the last treatment, but you should be able to kind of tease out the results at the end of this today, because we do have one that is just wet and dry, and then we have one that is heated and wet. So you should be able to see, if there's a huge jump in activity, like huge jump in CO2 with the heat, experiment portion, then you might expect maybe temperature has a bigger impact on microorganism activity. If there's not a huge difference between the cool and wet and the warm and wet, then you might say, moisture is probably the greatest factor in CO2 production, which means that it's the biggest factor in microorganism activity. So kind of think about what you might expect to see today, and we'll go ahead and show you how we measure these things. So for each of these chambers, we have a CO2 sensor hooked up to it, and we have these little computers that are meant to just work with these CO2 sensors. So they put off a graph, and we're allowing this to just kind of go for a few minutes without any soil in the chamber, so we get a good baseline just to make sure that our CO2 sensors are all working. So I've been letting that run for a few minutes, and what you should see, we'll see some up and down just because the sensors are not, top of the line, and there is going to be some changes just naturally occurring, but it should kind of basically be a flat line when you're using that with no soil inside the chamber, because there's, supposedly no microorganisms in there right now putting off any CO2. So what you're going to be able to do is have a graph from each of these, which would give you a slope that you can find by just highlighting a portion of that graph to see how much the CO2 changed. So we'll go ahead and we'll give you some baseline data with an empty chamber, and it should be basically flatline, so no change in CO2 throughout that. And we're going to go ahead and add CO2 to each of these 3 chambers and see what happens in each of our treatment types. So we'll go ahead and get that started, get our samples measured out. So we have one scale up here, and we're going to measure 100 grams, 100 grams of soil into each of our chambers. And what we usually, tell you if you were in the lab is we want to make sure that our CO2 sensor is not sticking inside of our little Ziploc container in here, because we don't want to be, covering up any of that CO2 sensor. So we're always careful about making sure that our soil is kept separate from our CO2 sensor. That's why we have the container inside of a container for this setup. So we'll go ahead and do that, and then we'll show you kind of the results at the end of it. Okay, so Lindy and I have gotten the first couple ready to go. So we have our dry soil on the end of the counter here, and we have our wet and cool soil in the middle. And then finally, we're going to add the warm and wet soil. For each of these, we want to make sure that our CO2 sensors are, approximately at the same level before we start, so we know that we have a solid baseline for all of them. And there is always some fluctuation with these things, but for the most part, they're all around 450 parts per million for CO2 before we get going. So we kind of had them all in the closed chamber just to stabilize and calibrate them. So now we're going to get started, and we're going to let them all run for 5 minutes. And so we're going to be recording data on each of these CO2 sensors for 5 minutes. So we're just going to get those all rolling at the same time. Lindy's going to get our final one weighed out here. So we're doing 100 grams of soil for each of these things, plus or minus 1 gram. So we don't have to be perfectly exact since we're using a pretty good portion of soil.
Maybe. There you go.
So that's 107. So we'll take, if you want to just take that little chunk off the top there.
There we go.
Okay, so we have about 100 grams of soil for each of them. And we have plugs for each one of these containers because we want to make sure that we don't have any of the atmospheric air getting into our containers. So they should be a closed system. And then on each of these, we're just going to, we have this graph function on all of these, and we can just hit go, it starts, and it just records until we tell it to stop.
So we just ran the experiment for 5 minutes with our CO2 sensors for each of the 3 different treatments. And then the way it worked was it measured the CO2 levels, parts per million, and then graphed it in comparison to the time. And so you'll see kind of a slope here, and this is for the cool and wet treatment. And so it graphed in CO2, and it kind of has this upward trend. And then once we finished the experiment, then we were able to get the slope and the line. And so for this treatment specifically, our slope ended up being 0.068. And so that's showing that there was an increase of CO2 over time. All right, so we ran all 3 of our treatments, and we have all of our graphs set up here. And so after running them, we made sure all the graphs were to scale. This is important, so make sure we're comparing the same data and same information. And so you can really see kind of what happened with each treatment. And so for this first one here, this was the cool and dry soil. You can see the graph is fairly flat-lined, not a big slope. And our slope actually came up to be 0.003, which is a pretty small value. This isn't surprising since we didn't really change much to the soil. It's just cool, dry, just how it's been. So this was as expected. For the next treatment, for our cool and wet soil, you can see there's definitely a little more of a slope here on the graph, with a little bit of an increase in CO2 parts per million. And then this slope ended up being 0.06, so a little bit bigger than our cool and dry soil, but still not a huge increase. And again, this is kind of what we expected. So we changed one variable, added some water, so it's a little different, but not drastically. And then for this one, this is our warm and wet soil. And you can see here, there's a much bigger slope than the other 2. And then the slope value actually came to 0.14. So this is larger than the other 3, which is, again, kind of as expected because we added the heat and the water to the soil. So, this is suggesting that this has increased the microbe activity levels, and they're producing more CO2 with those 2 variables.
Alrighty, so we have our 3 treatment types, and we got data for each of those. But if you were in class, you and your partner would be doing this, and everybody else in the class would be doing the exact same thing. And we've reiterated several times in the semester that sample size is important. So we want to do this more than one time, right? So don't just use the data that we gave you right here. We wanted to show you kind of what the raw data would look like if you were doing the experiment yourself. But now you need to go in on Canvas and get the class data that Dr. Dr. Robbins has posted for you there. Make sure you go in and watch the lecture video. Again, this was just a demonstration of how would you be doing the exercise yourself. You can get everything else you need from the lecture video and from that PowerPoint to then fill out the questions in your manual. You will have to make a graph. So just make sure you kind of understand that we started with that baseline, which should have just been like a really flat line because we just had an empty container with a CO2 sensor in it, which is basically 0.00005. And so then we had a slight increase in our cool and dry, a bigger increase in our cool and wet, and then an even bigger increase in our warm and wet. So think about what those things mean in terms of microbial activity, because that's what this all comes back to. There are living things, bacteria and fungi in that soil that are actually producing CO2, just like you do when you're going about your normal day and breathing out CO2. So think about what all that means. Make your graph based on the class data, not on the data that we gave you here today, and let us know if you have any other questions.